Thank you, all of you. Welcome for the biology classes, dear student. We have studied in molecular basis of inheritance so many concepts. You know that there will be a structure of DNA, component of DNA, experiments concerning with DNA as genetic material, packing of DNA, and also replication of DNA, semi-conservative replication of DNA, and experiments concerning with this replication of DNA, and also central dogma of molecular biology. It is very important. According to central dogma of molecular biology, the DNA is copied into mRNA and again mRNA translated into particular protein as the guidance of DNA master molecules as the language depicted in the DNA. Proteins. Protein is the polymer. Several amino acids are joined to form a protein. Here, we shall move on to the genetic code. After transcription, transcription contains transcription unit and transcription prokaryotes and transcription in eukaryotes. Prokaryotes and eukaryotes, both. And lastly, next we will move on to the genetic code. You know that the DNA contains DNA is a double-stranded structure, polynucleotide chain. This DNA, there will be a language. It contains genes. You know that gene is the functional unit of heredity. It is the functional unit is a structural gene, cistron. It contains cistron, polycystonic and monocystonic, monocystonic. The cistron is the undergoes mutation, muton, and also recombination. Racon contains mutons and recons. The fine structure of DNA. In the DNA, that is, the genes are present in the coded language. You know that the nucleotide or base pairs of DNA nucleotide and RNA nucleotide. DNA nucleotide, suppose A, G, T, C, A, G, T, C, A, T, T, G, C. This is the one strand of DNA. It is called as a template strand, antisense strand. This the complementary base pairing takes place. A will T, C, T, A, C, G, A, again T, T, A, G, C, and C will complementary to the G. Therefore, this is the language present in DNA. But in transcription, mRNA is synthesized by mRNA polymerase and it will come to the starter initiation point. Sigma material accepts and starts the transcription. RNA polymerase synthesizes the short stretch of mRNA copy by this DNA language in this own form. It is converted into T is converted into uracil, language of mRNA, and later on the three codons according to genetic code will be transcribed and translated into different proteins. Proteins after completion, it will pass on to the cell. Thus, the protein synthesis is takes place. Protein protein synthesis will take place. Here, the coded language depicted in DNA is called as genetic code. Genetic code. The coded language. The coded language. Coded language defected in DNA. The coded language defected in DNA is coded language defected in DNA is called genetic code. The coded language defected in DNA is called as genetic code. The coded language defected in DNA is called as genetic code. This genetic code, it is a language of DNA. It is a language of DNA. It is a language of DNA. Suppose the DNA contains many coded information. It is a language of DNA and is a set of 3 nucleotide bases, nitrogen base, and it is a set of 3 nucleotide bases. It is a set of 3 nucleotide bases. It is a set of 3 nucleotide bases. And on DNA arranged in a specific sequence. On DNA arranged in a specific sequences. Specific sequences. Genetic code, the coded language defected in DNA is called as genetic code. The coded language defected in DNA is called as genetic code. It is a language of DNA. It contains the AGTC, etc. The genetic code. It is a set of 3 nucleotide bases on DNA arranged in a specific sequence. And any living cell is made up of. Any living cell is made up of. There will be a 70 to 90% living cell made up of 70 to 90% water, 70 to 90% water, and also 17 to 18%, 19%, 17 to 18% nucleic acids. Nucleic acids, Na, aerated Na, nucleic acids. Nucleic acid, and 5 to 7% carbohydrate, 7 to 5 to 7% carbohydrate, and 2 to 3% fat, 2 to 3% fats, and only 1% mineral ion. And what are nucleic acids, carbohydrate, fats, etc. Here is the protein, is the nucleic acid structure contains very much quantity. Therefore, protein is very important and essential material. Protein is essential material, and this is the not rigid, but it may be a chain. Almost the protein is very important. From this, we can, dear student, will come. You will come to know that protein is essential material. Protein is essential material, and the constitution of protein. You know that the what is the protein contains constitution of protein. Protein is the made up of many amino acids. Amino acids are polymer, and amino acids are monomer. Several monomers. Suppose the component of protein made up of amino acids. Amino acids. Amino acids. Amino acids. Protein contains amino acids. You can discuss about the protein is the bead on strings. The structure that is the bead on strings. This is one molecule of protein. Protein contains, this is the amino acid 1, amino acid 2, amino acid 3, etc. There will be 20 amino acids are bonded by polypeptide bond. Polypeptide bond. Peptide bond. Protein is made up of many amino acids. These amino acids bonded by peptide bond forms one protein. Therefore, this way the protein will be formed. Therefore, the protein is the essential element. Several amino acids, say A1, A2, A3, etc. A 20 amino acids make one protein molecule. Therefore, in this, there will be a genetic code is a set of symbols or words. Genetic code is also a set of symbols, words or letters used in represent coding and decoding. It is a set of words, symbol used in coding and decoding. Coding, it is very important. Coding and decoding. Decoding means AUG is the one code. It is symbol. It is symbol. It refers to the methionine. Methionine is one of the amino acid. Methionine is amino acid. There are 20 different types of amino acids are present. Arginine, aspartic acid, glutamic acid, etc. Many phenylalanine, leucine, isoleucine, and methionine, start codon, valine, serine, proline, threonine, alanine, and tyrosine, glutamine, arginine, lysine, aspartic acid, glutamic acid, etc. Arginine, serine, and lysine. There are many 20 amino acids present in the cell. Therefore, it requires coding and decoding. In computer language, there will be writing how the coding of these and decoding. Elementary work be done by coding and genetic material coding ಮತ್ತು ಡಿಕೋಡಿಂಗ್ ಸಾಫ್ಟ್ವೇರ್ ಇರುತ್ತೆ ಕಂಪ್ಯೂಟರ್ ಅಲ್ಲಿ. ಅದರ ಲಿವಿಂಗ್ ಆರ್ಗಾನಿಸಮ್ಸ್ ಹೇಗೆ ಸೃಷ್ಟಿ ಮಾಡಬೇಕು ಅಂತೇಳಿ ಅದರಲ್ಲಿ ಇರುತ್ತೆ. ಅದರ ಮೂಲಕ ಇವುಗಳನ್ನ ನಾವು ಅರ್ಥ ಮಾಡ್ಕೊಳ್ಳಿಕ್ಕೆ ಅನುಕೂಲ ಆಗುತ್ತೆ. Again, it also describes the relationship between four types of nucleotides. Four types of nucleotides. Therefore, this DNA contains AGTC, AGTC. The complementary one is ATGCT, AC, GATGC. This is the complementary strand. By this, there will be a mRNA copied a template strand. It will convert it into A U G C T A C G A U. Here, instead of T, there will be a U. This is the coded converted by mRNA. This will be shows that the three codons makes this coded language. It refers to the different types of amino acids. Different types of amino acids. Therefore, ಇದನ್ನು ಹುಡುಕ್ತಾ ಸಿಕ್ಕಿ ನಮಗೆ genetic code. Genetic code is the four language. It contains four language. A, G, U, C. AGTC in DNA and AG, UA, UCAG in RNA. The different codon systems are as follows. At the first time, Dr. George Gamow physicist grouped these codons into different groups. Singlet codon according to him, doublet codon and triplet codon. Therefore, some scientists help or undergoing corona makes these RNA synthesize RNA molecule in laboratory condition, and later on Marshall and Nirenberg obtain the cell-free proteins, and also they find out that Severoaceae protein polynucleotide polymerase. In this DNA genetic code contains. And also next the genetic code. Different codon systems are as follows. Different codon systems. Different codon systems. Say, first one is the singlet codon. Singlet codon. What is mean? 20 amino. Then there will be a 4 nucleotide. There will be 4 nucleotide. Each nucleotide refers to one amino acid. There will be 20 amino acids. 16 will be removed. 4 nucleotide nitrogen bases A, G, T, C, etc. That's what we amino acids are 20 amino acids are present in the cell or body contain 20 different types of amino acids, and this will remaining 16 will remain. Therefore, this concept was not accepted by the science. Second, there will be a here is the UGAC. This type is UGAC. mRNA. Codons is reference to the mRNA. U always instead of T there will be a U. RNA language UGAC. By this will not accepted. And second one, the scientist made doublet codon. There will be 2 nitrogen bases codes for 20 amino acids, 4 codes remain. So thus the codon will take the charge. First UC AG, here the UC AG. UC AG, 16 codons will be formed. 16 codons, these are also discarded because of there will be 20 amino acids. Therefore, again lost the third system is the triplet codon. Triplet codon. Triplet codon will be succeeded, and nowadays we use this for study of them. This type of triplet codons will be used by the scientists. According to this, the construction is very simple. There will be a 64, 4 into first 4 raised to 4, 4 raised to 2 into 60, 4 raised to 3 is 64